Hello everyone, welcome back to Chemical Engineering and Aspen channel. As you know that from past few months we are focusing on the module of chemical reaction engineering and we have so far presented 39 videos for our valuable viewers. In today's video, which is our lecture number 40, we will be focusing on the differential method for collection and analysis of rate data. So far we have studied three methods: integral, method of half-lives, and method of initial rate. And method of excess was also studied, but that is again included in these methods. And today we will study the differential method, and among which we will study the numerical method. However, this lecture is divided into two parts. In this part, which is presented to you, we will study the theoretical concepts and will solve the numericals initially, and then in next lecture we will apply the numerical method in detail. So talking about the differential method of analysis, let's assume a reaction which is proceeded isothermally in a batch reactor having constant volume, and the change in concentration of A is recorded as a function of time. So if we apply the mole balance and rate law concepts and combine these, we get minus dCA over dt is equal to K of A Ca raised to power alpha, where this alpha is the reaction rate constant. And if we take natural log of it, we get natural log of minus dCA over dt is equal to alpha natural log of Ca plus natural log of K of A. And this becomes the straight line equation y is equal to mx plus c. It means that we have to draw a plot between natural log of Ca and natural log of minus dc over dt, and the slope of the line will be alpha, as you can see in this diagram as well, that we have drawn the plot between Ca and minus dc over dt, but we have taken the log plot. So accordingly, the slope of the line will be alpha, which is the reaction order. In another way, what we can do, we have to first choose a concentration in the plot, let's say Ca at some point P, and accordingly we have to find minus dc over dt at that point. Accordingly, these may not be the data point, it just need to be on the line, and then we have to simply divide minus dca over dt at some point divided by cap raised to power alpha. As you can see this diagram, that this plot is drawn, the straight line is obtained. We have to just select a point from here, go to this line, and then we select suitable value. This value divided by this value raised to power alpha will give the answer of k. So obviously in the first step, we will find the value of alpha, and then in the second step, we will find the value of k here. The slope of the line will be alpha, and accordingly we can find k from this equation. To obtain the derivative, now this is the main thing, that we have to calculate the value of dc over dt. We have to use different methods: graphical differentiation, numerical differentiation, or polynomial fit. And today our focus will be on the numerical differentiation. Now, how this method is proceeded, the numerical method of differentiation, let's assume that we have been given the time versus concentration data at 5 different points. And if we include the initial point, it will be the 6 different times. And accordingly, we are given the different values of concentration. For T naught, the value Ca naught, T1, Ca1, T2, Ca2, up to so on T5, Ca5. And at each time, we have to calculate dCA over dt. It means we have to calculate it at 6 points. The first point and the last point, if we just take it out from the equation, we get the 4 points: T1, T2, T3, T4. This may not be same every time, so we have to adjust the equations. But now how we can proceed with it, that dC over dt at time t is equal to 0 is equal to minus 3 times Ca naught, which is the first value, plus 4 times Ca1, which is the second value, minus Ca2, which is the third value, divided by 2 times delta T. And this formula will remain the same irrespective of how many points are involved in the system. In the same way, if you look at the last point, dC over dt T5, this will obviously depend on how many points are involved, because it takes into account the last 3 points, which are Ca3, Ca4, and Ca5. So Ca3 minus 4 times Ca4 plus 3 times Ca5 divided by 2 into delta T. So the first parameter, the initial point, takes first 3 points, and the last parameter takes last 3 points. So if there are, for example, 10 points, Then the last parameter will take point number 8, 9, and 10, while the initial point will always take 1, 2, and 3. For the rest of the points, which are between initial and final points, this is the generic formula dCA over dt. Pi is equal to 1 over 2 times delta t, bracket CAI plus 1 minus CAI minus 1. It means that if the value is calculated at T1, it will be Ca2 minus Ca0, as you can see here. Ca2 minus Ca0 over 2 delta t. For T2, Ca3 minus Ca1 over 2 delta t. For T3, Ca4 minus Ca2 over 2 delta t. For this T4, Ca5 minus Ca3 over 2 delta t. And if these points are reduced, accordingly the formula will change. So if you can understand this concept logically or theoretically, then you will be able to solve any numerical irrespective of any number of points. You have to just remember that the first point contains first three concentrations, the last point contains last three concentrations, and to find the intermediate points between the initial and the final points, then you have to calculate the above and below value. For example, for point 1, you have to calculate the concentration at point 2 and concentration at point 0. And in the same way, you can calculate it, and you have to kept one thing in mind, that this value of delta t should be same. It means that the independent variables are equally spaced and independent variables are time in the system. Now going to numerical, we have already solved the numerical in integral method, that is the same numerical that time versus concentration data is given to us in a system of triphenyl methyl chloride trityl and methanol system. Methanol is in excess, as we have seen earlier, and we have been given the concentration from 0 to 300 minute with a time interval of each 50 minutes, 50, 30, 30.6, 25.6, 22.2, 19.5, 17.4. We will just today develop an algorithm, and in the next lecture we will apply the numerical method. So this is the data which is given to us, that you can see that with the increase in time or as the time progresses, the concentration of A is decreasing in the system. Now, if you remember the algorithm for data analysis, the first part was to determine which of the model to be used. Since it's an homogeneous system, so we will apply the power law model, which is minus Ra is equal to K of A, C raised to power alpha, Cb raised to power beta. The type of reactor, obviously it's a batch reactor. The measured variable is concentration, and if there are any assumption in step 4, yes there are, that methanol is in excess. So accordingly, that Cb will become equal to Cp naught. Once you combine these, you get an pseudo rate constant, which is K prime Ca raised to power alpha. Going to step 5, which is the algorithm for CRE, which is mole balance for batch reactor. DNA over dt is equal to Ra into V. Rate law is minus Ra is equal to K prime Ca raised to power alpha, as we have developed. Then in stoichiometry, we say that it's a constant volume system, so V is equal to V naught, Ca is equal to Na over V, Na over V naught. Then we have to combine all these. We get minus 1 over V dNA over dt is equal to minus Ra, and accordingly this becomes minus dC over dt is equal to K prime into Ca raised to power alpha. So we have developed all these equations specifically in step 5 earlier in our previous lectures as well. So going to step 6, we have again written that equation. We have to simply take the natural log, and accordingly we get natural log of minus dC over dt is equal to natural log of K prime plus alpha natural log of Ca. Now that corresponds to y is equal to c plus mx, where m is alpha, x is natural log of Ca, y is natural log of minus dC over dt, and the c will be natural log of K prime. And we have to use the differential method to find out the value of alpha and K prime, and from K prime we can determine the value of K. And we will be using the numerical method in the next lecture. So I hope you have understood all the aspects of this lecture. If you have any query, feedback, suggestion, please provide it in the comment box, and I would be happy to answer. So that's it from today's lecture. Thank you so much. Please do watch, like, share the video, and subscribe to the channel. Also click on the bell icon to get all updates related to this channel. Till then, it's goodbye. Stay tuned for more exciting videos on this channel.